Hello everyone, my name is Ashna, and in this Chalk Talk, I will be discussing the supramolecular assembly of peptide amphiphiles. To begin, let's refresh on our basics. Peptide amphiphiles are small molecules that contain hydrophobic components bound to peptides. The peptide sequences in these molecules are fairly short, right about or under 10 amino acids, and they are linked to an aliphatic tail consisting of 10 or more carbon atoms. The short peptide moiety forms beta sheet structures, whereas the aliphatic chain moiety self-associates via hydrophobic interactions. The location of sequences within the molecule itself is important because beta sheets can form among the amino acids near the alkyl tail and also the residues furthest from the tail, which can then be charged to promote solubility. Therefore, depending on the amino acid sequence, peptide amphiphiles have the ability to self-assemble into a variety of nanostructures, including, but not limited to, micelles, vesicles, bilayers, and nanofibers. In water specifically, beta sheet formation and hydrophobic collapse of the aliphatic tails induce assembly of the molecules into long cylindrical or ribbon-like nanofibers. Here I've included a modified peptide amphiphile, which includes a charged group as well as a bioactive epitope specific to an antibody in a particular field of research. The final structure being a nanofiber. Supramolecular assembly of peptide amphiphiles relies on protein assembly pathways because these pathways are critical to correct folding and function of proteins. The main way that this is done in these amphiphiles is by hydrogen bonding. The primary sequence of amino acids present in each molecule utilizes hydrogen bonding as a fold into secondary peptide structures, which include alpha helices and beta sheets. Alongside hydrogen bonding, hydrophobic interactions are also integral to the formation of these assemblies. Increasing the hydrophobicity of a molecule by decreasing the hydrogen bond forming ability of solvents directly leads to aggregates. Put this molecule in a polar solvent and now you have a micelle. What if your peptide moiety has additional non-covalent interactions? Now you have a cylindrical micelle. The important difference between the two is dependent on how many non-covalent interactions are taking place within the peptide moieties. These include everything that we've talked about this semester, mainly the dipole interactions and pi stacking to the applicable amino acids, such as tryptophan, phenylalanine, and tyrosine. Here is an example of how polar and non-polar solvents can affect peptide amphiphile assembly. As on the left here, water allows for the formation of a bilayer with the hydrophilic peptide moieties facing the solvent. On the right, the opposite is true, since tetrachloromethane allows for the flip formation to occur with the hydrophobic moiety facing the solvent. On the other hand, pH can also have an impact on the formation of structures, especially with modified peptide amphiphiles including charged groups. In this figure here, nanofibers form when pH is greater than the pKa, but changes in pH are affecting the ability of the nanofibers to bundle. With strategic design, peptide sequences can be placed in a manner where they can attract or repel other peptide amphiphiles. This self-assembled nanofibers bundle due to interactions with neighboring nanofibers. Here, the design achieved a pH-responsive system via charged residues arranged in a complementary attractive fashion, resulting in the bundling of nanofibers, and those in a complementary repulsive fashion, which results in dispersed fibers. Down here, we have a tubular structure assembly in which alternating D and L amino acids produce a flat ring-shaped structure. In a polar solvent that promotes hydrogen bonding, these rings can assemble into stacks. If you notice around the perimeter of the ring, there are many residues present. During strategic design, these residues can be made hydrophilic or hydrophobic, both of which affect the final function. If the residues are hydrophobic, these tubular stacks would integrate nicely into the lipid bilayer and act as pores which can transport hydrophilic substrates through. If the other way around, the opposite becomes true because that assembly would be surrounded by a polar environment and it would be able to transport hydrophobic content where need be. With just simple tweaks to the amino acid sequence, the ring size can also be altered, making the pores more selective. Now, let's take a look at a ternary phase diagram to understand the interactions more. Here, our three players are DPPC in yellow, which is a phospholipid with a polar head group. DSPE PEG in green is a polyethylene glycolylated amphiphile, and our peptide amphiphile here in blue has the characteristic dense hydrogen bonding section right here. In this phase diagram, it is clear that upon introduction of peptide amphiphiles to the lipid mixture, the self-assembly phenomena is enriched significantly. Through the hydrophobic interaction of the alkyl segments and hydrogen bonding among peptide segments, beta sheet structure is more favorable. The amount of the peptide amphiphile plays an important role in deciding the morphological transformation. With more peptide amphiphiles present in the system, rod-like micelles are likely formed, and with a lower amount, the molecules tend to self-assemble into spherical micelles. Now that we understand how hydrogen bonding and hydrophobic effects can drive assembly, what is the reason why? We can take a look into the thermodynamics of these assemblies to understand. Peptide amphiphiles spontaneously form supramolecular assemblies, and to understand spontaneity, let's revisit Gibbs free energy equation. Spontaneous reactions require low enthalpy and high entropy. Having a high delta H is energetically unfavorable, and combined with low delta S, which is entropically unfavorable, we have a non-spontaneous reaction, which is unlikely to proceed by itself. It is possible to be energetically unfavorable and entropically favorable, and this leads to a spontaneous reaction at high temperatures. More importantly, it is also possible to be energetically favorable and entropically unfavorable and result in a spontaneous reaction at low temperatures. Supramolecular assembly is driven by an increase in entropy at low temperatures and decrease in enthalpy at high temperatures. More interactions between different moieties contribute to the thermodynamics of the assembly by decreasing overall enthalpy. On the right here is detailed how exactly entropy is increased during assembly. In a monomer system, these molecules are floating about and trapping water in between them, seen here. This is entropically unfavorable since the water molecules are not free to move as they please. With the hydrophobic effect, these molecules assemble in a way to avoid contact of hydrophobic moieties with water molecules. What this does is release entrapped water to the surroundings and increase entropy within the system. Here is a simplified figure of this effect. It shows that on the left, delta H and delta S are negative, and delta G is positive, indicating a high energy state of these molecules that are not permitted to flow freely. On the right, after the hydrophobic effect takes place, we can see delta H and delta S turning positive and delta G turning negative, which indicates a low energy state of the molecules, which can also flow freely. Finally, let's take a look at the thermodynamics of protein folding, the template from which peptide amphiphiles assemble. Here I've drawn out a protein folding landscape. There are two different types of interactions occurring here. There is intramolecular and intermolecular interactions. Let's walk through this then. Up here at A, what we have are the unfolded proteins. These are the most energetically unfavorable form of proteins, thus leading to the following forms. B is the more stable form and is known as the folding intermediates. You can see that here. Here is when hydrogen bonding comes into play, and we see our beta sheets and alpha helices. C is further folding into the native state of the protein. All non-covalent interactions occur here, but mainly hydrophobic interactions, which produce an entropically favorable state. D is consisted of partially folded interactions, partially folded states, which are more stable than the folding intermediates but less than the native state. Moving on to the right half are the intermolecular interactions. E is consisted of amorphous aggregates, more specifically described as a mess of many interactions. F is consisted of oligomers, which are slightly more stable than the partially folded states at D. Finally, we have the most favorable form of folding. These are the supramolecular assemblies. Again, these can be in any form, but they are the most stable form of protein folding themselves. Overall, what is important to remember about peptide amphiphile supramolecular assembly is that there is no one-size-fits-all, and these are incredibly tunable molecules that lead to numerous forms of assemblies. The two main interactions to keep in mind are hydrogen bonding and hydrophobic interactions, which then affect how the molecules interact with specific solvents and how they aggregate. I didn't cover a lot of the applications for these amphiphiles, but they are very popular in biological studies since the peptide sequences are bioactive and biocompatible. With that, I have concluded my topic. Thank you for giving me your time, and please let me know if you have any questions.